Our last speaker for this session is Cam Bielaski, Neural Networks in Hierarchical Neural CA.
All right. Thank you. Yeah, neutral genotype networks in hierarchical neural cellular automata. Um, I'm sure many people made the mistake of thinking that was actually neural networks, but no, neutral genotype networks, and that's where I'm going to start. So when we talk about evolution, there's this kind of tension between evolving organisms where if I'm an organism and I want to, compete, I want to preserve my current adaptive functionalities, but I also need to be able to innovate and find new innovations to be able to compete. And it's kind of this evolutionary game theory arms race, right? Um, uh, so yeah, there's this tension here between finding innovations and also being robust. And so if I want to change my system structure, I don't want to lose my current functionality when I'm exploring the functions, right? So uh, the kind of synthesis that addresses that tension is this idea of neutral mutations, neutral networks. And there's this debate in evolutionary theory between selectionism and neutralism, where in selectionism, all mutations are strictly good or bad, or this idea of neutral networks and neutral mutations kind of proposes that evolution proceeds by way of traversal across neutral genotype networks, where you make many mutations that are effectively neutral in your fitness, but um, this traversal of this neutral network gives you more opportunities to find an innovation. So I think the best way to talk about this is by giving an example. So here's um, a genotype phenotype mapping. So we have a sequence of nucleotides, right, that get mapped to an RNA secondary structure, which is associated with its function, and so therefore it's kind of fitness, right? And so you can have um, sequences of nucleotides um, that are different, that get mapped to the same RNA secondary structure. So this is a very simple example where uh, one nucleotide is changed, but the secondary structure and thus the function is unchanged. So you can get these networks of genotypes that all get mapped to the same uh, phenotype. And so, yeah, we call it. I've been saying this word neutral genotype networks, and that's what this is. And one of the consequences of this is that you can have genotypes um, that look very, very different if you were to just look at the actual the genotype encoding itself. But um, when you actually run it through the genotype phenotype map and actually express the phenotype, uh, you get effectively the same thing. Um, so there's a lot of implications of this, and these have been really well studied in biology, uh, mostly by Andreas Wagner. Um, and so these occur at many different scales, right? So the simplest example is like a codon getting mapped to an amino acid, right? You can there's 64 possible codons, and they only get mapped to 21 amino acids, right? So there's already neutrality at that base most level in biology, but there's a bunch of empirical evidence that uh, that these neutral genotype networks exist at many different levels of organization. Um, and my intuition, I kind of put an arrow above this. Uh, my intuition is that there exist a lot of these neutral networks across many different scales. Um, and so I think another way to think about this is the evolution of sort of error correction. Um, Error correction mechanisms to preserve to preserve adaptive functionality. And yeah, yeah. So yeah, this work kind of investigates or uses this idea and investigates whether or not we can design artificial systems so that they support and exploit these neutral networks. So the model that we actually evolve here is a hierarchical NCA. So the way this system differs from a traditional NCA is that you have grids that are many different levels of resolution that are all interacting with one another. And so you have a cell that not only integrates information from its local neighborhood, but also integrates information from the grid below it and the grid above it. So this is kind of inspired by the multi-scale competency architecture, right? In biological systems, we have, evolved structures at many different scales, and they give rise to multi-scale organizations. And so each layer of this system has its own kind of genotype, which is defined by a very minimal neural network. And so this is kind of in contrast to a single scale NCA where all of the dynamics are emergent from a single scale. And so another way of thinking about this is providing evolution a scaffold for evolving dynamics at many different scales, and evolving them directly instead of sort of forcing them to emerge from a single scale. Okay, so when we evolve these systems, we take the top layer and we binarize it. We take the values at the last few time steps and we binarize them into zeros and ones, and then we have target shapes that we're trying to match to at the end of 100 time steps. So this is basically kind of a morphogenesis and homeostasis task, if we can use the biological analogy. And so this is the genotype we're working with across all of the different scales. And so, yeah, you have parameters associated with each level of resolution, which we will talk about later. So here's a snapshot of one kind of in action. You have these signaling channels across 4 different scales, and then we take the values of the top layer, binarize them, and compute the loss. So again, we have the genotype, and then, we take the last 5 time steps, the binarized phenotype, we compute the Hamming distance away from the target shape, and we get the loss. And so here, how we actually measure neutrality is by looking only at the loss, so this is like the evolutionary fitness in evolutionary terms. And so you can have many different genotypes that get mapped to the same loss, and that's thanks to the kind of the binarization step. So I look at different connection structures. I won't talk about this too much, but I look at local connections where cells are only integrating from the cells directly below it and the cell directly above it, and in a bidirectional way or in a unidirectional way where cells only look at cells directly below it, right? So bottom left. And then I look at non-local and so non-local connections where I take the connections that would normally be connected to cells directly below it or above it and randomize them to a random scale and a random offset from the cell. So you have these sort of long range connections. And then, right, I won't talk too much about the unidirectional versus bidirectional. I'll leave that at that. And so here's kind of the fitness of these systems. The first thing I want to point out is that the non-local connections outperform the local connections. My intuition for why this is basically these long-range connections are turning these systems into like small world networks, or it's making it more brain-like where you have these long-range connections and it's more optimizable. And then you have like a slight, so the green being slightly better than the red. You have a slight increase in performance of the unidirectional. So that, yeah, this is the fitness of these systems. But if I want to zoom in on just one of them and look at the mutations over evolutionary time, you can see over evolutionary time, there's you have about 25%-ish of the mutations that are happening are neutral. So neutrality, neutral mutations are sort of built into all of these systems. And then on this right side here, so yeah, I should mention this is averaged over 500 hill climbers. But if you see at the right side here, each of these points is how many steps along a neutral network an individual took before finding a beneficial mutation, right? So if we look at this guy, this particular HNCA took 1,400 steps along a neutral network before finally finding a neutral mutation. And so I think that's quite interesting and could inform how we actually design systems. And then, so now I looked at the neutral mutations across these different scales and looked at how that changed over evolutionary time. I won't linger too much on this, but so, right, so you have basically differential neutral mutations across different scales, and that differential neutrality is preserved over evolutionary time. Okay, yeah. I'll say something real quick about this. So you have a few different. Oh man. Okay. So this looks. This is just a normal 64 by 64 NCA. This has 9 parameters. It's basically just a little convolution kernel that updates. And so this is non-hierarchical, but if you tweak a parameter, which is the threshold of the activation function, you can actually use it as kind of a knob for how much neutrality there is. And you can see there's differing amounts of evolvability. And I am kind of claiming that this is basically changing the structure of the neutral genotype network of the system. So we get this kind of, what the neutral genotype network kind of theory produces is you have individuals that get increasingly fit by trying stuff out at these lower scales, and then there's sort of an evolution of error correction mechanisms, which which allow this experimentation at these lower scales, right? And so this is kind of how evolution proceeds. And then finally, another kind of interesting observation that we do also see in nature is that we can see these morphogenesis trajectories diverge from each other. So these three rows that you see are three HNCAs all connected by the same neutral network, but they're spread out a little bit. So they, when you actually play out their dynamics, they diverge, but they end up at the same place. So they end up at the same place where the phenotype is ultimately evaluated. And I think that that's also been observed by Andreas Wagner in biology, where you have kind of many different ways of making the same body. So yeah, in some, you can build NCA architectures in many different ways, and I would argue the changes in the architecture you can think about as changing the neutral genotype network structures, which change the evolvability of the system. And so observing the evolutionary dynamics across these neutral networks can explain and inform system design. Thanks.